Hello, everybody. Welcome. Thank you very much for attending today's Precisionary Instruments webinar. I'm Abby Chu, Chief Scientific Officer here at Precisionary, and it's my pleasure to host this session. And today we're honored to feature Dr. Jian-young Wu, Professor of Neuroscience at Georgetown University Medical Center, and you'll see his beautiful picture of the campus right there. Dr. Wu has been a pioneering figure in experimental neuroscience for over 3 decades, focusing on large-scale neuronal activity and the dynamics of brain network oscillations. His lab's work with ex vivo hippocampal slices has provided powerful insights into the mechanisms of memory, neurodegeneration, and drug discovery. So in today's talk, Dr. Wu will walk us through the generation and significance of hippocampal sharp waves, demonstrate practical methods for preparing high-quality slices using the compressed-tone vibratome, and share examples of pharmacological modulation relevant to neurodegenerative research. So again, thank you for joining us today. Please feel free to submit your questions and comments to the chat throughout the presentation, and we will address them both during the Q&A at the end and also during Dr. Wu's talk. So with that, it's my pleasure to welcome Professor Wu.
Yeah, hi. Hello, everyone. It's a pleasure to give a talk, give a webinar about my work. And most importantly, I would like to keep the lecture more emphasized on the methods, how to get a good slice, how to get a good sharp wave ripples. And so my talk will be, hold on, let me try to advance my slides again. Okay. So about hippocampal sharp waves. So my talk will be divided roughly in 3 parts. One part is what is a sharp wave ripples and why it's important for sharp wave ripples in the brain slides to help our neuroscience research and brain degeneration. And the second part will be a method and how to use compressed tone to get a good slides for obtaining good sharp wave ripples. And third part, I will show a few examples of research in my lab. So we know the hippocampal sharp waves are a conserved neural activity all over mammalian animals. And possibly, if you go down to the evolution line, it probably can go down to reptile, even frogs and the fishes. There are papers about fishes have sharp waves. So that's conserved activity means must be some function that is important. And what is a sharp wave? So actually sharp wave means a field potential peak, voltage peak, with a duration about 50 millivolts. And the amplitude is pretty high. And according to some people, they said it's like the largest amplitude activity is epileptic spike. The second largest is a sharp wave. That's about 5% of all neurons or have active in a short period of about 50 milliseconds. And the ripple oscillations are fast oscillations accompanied with a sharp wave. So those ripple oscillations are generated by PV inhibitory neurons. Majority of these neurons are probably positive PV neurons. And these neurons, when a lot of pyramidal cells are firing, so those neurons are highly excited and they generate a rhythm of its own network. So those called ripple oscillations, about 100 to 200 hertz. Okay, so why is it important? So people believe that sharp wave is essential for memory consolidation. So those are related to a theory we call a two-step memory theory. So the two steps means there's an online state and it's an offline state for memory. and memory consolidation. So the online is for registering memory. So during the behavior, exploring or running are the online state. So in the hippocampus, mostly we started in rodents. So during the exploring and running, the hippocampus field potential are theta waves. That's a low, well, it's like a 4 to 7 hertz oscillations. And the spiking, those cells, CA1 cells are spiking, and those spikes are related to the spatial location of the animal. So this is called place cells. And offline are during sleep, non-REM sleep, or consummatory behaviors like they sit still or not moving. And so at this time, the theta wave disappears in the CA1 in hippocampus, and instead they have this sharp waves is like a 1 per second, accompanied with the rippleations. And during those sharp waves, the place cells, the sequence of the place cells will reoccur. So the sequence means if you go to an IKEA store, first you start in the kitchen section, and then to the bed section, and then to the office section, right? So their neurons will register your location in the IKEA store, and your sequence from the kitchen, bedroom to office area. So those place cells will fire in such a sequence, and these sequence will replay when you are in the non-REM sleep. So this is like a remarkable ability for mammalians to remember things. So imagine that if I ask if you remember what did you do last weekend, so most of the people will think, oh, I went to a movie theater, I watched this movie, or I went to a store, I went to a coffee shop, we did this, we ordered this. So these are like you experience only once. How can these memories become, like a lot of them become permanent memory in your brain? So the two-step memories are basically say, during the offline, during your sleep, those sharp waves will replay those sequence of place cells. Yeah, place cells is a definition for rodents. And actually the place cells are also in the higher mammalian can be, we call it concept cells. Let's say in the social relationship, like in bats or in the other social animals. And in humans probably also register the group in the members in the social group. And those concept cells will form a relationship. And during the sharp wave ripples, those relationships or the sequence of place cells will replay like a videotape once every second, and have a little piece of your memory registered during the day and replayed during the night. Okay, so there are other functions, more speculative. It's the second half of the sleep during the night. So the second half of the sleep, so those sharp waves doesn't have many sequences. So, but they do have other sequences, like they are predicting what will happen next day. And those we call the pre-play in behaving animals. And another function of those second half sleep sharp waves are they're erasing LTPs, erasing the memory already moved to the cortex. And then they give a space for hippocampus for the next day to prepare for registering new memories. So if we don't have a good sleep, we will experience bad memories the next day. Probably have some related to not washed out. Those old LTPs are still there, and to interfere with the new registration of new memory. Okay. Then in the ex vivo brain slides, if you keep enough hippocampal tissue, those sharp waves will spontaneously occur in the frequency similar. Can you guys see my cursor on the screen? I have a mouse cursor on the screen. Can you see it? Never mind. So in the ex vivo brain slides, on the top trace is a local field potential recording. And you can see the sharp wave once a second or so. And sometimes they go with a burst, several of them. And they're large, the amplitude are sometimes larger, sometimes smaller. And with a distribution, almost, we don't know what the amplitude means, but it's like a similar to a random distribution of amplitude. And in the signal, if you digest the signal, like I digest one sharp wave signal in the field potential, labeled by this green shadow, and you can see when I filtered 0.1 to 30 hertz, they only have a local field potential amplitude, or like a local EEG amplitude, pretty large, about this, in this example, about 500 microvolt, half millivolt. And then if I filter in the gamma frequency range, gamma is like 20, 40 hertz, like 30 to 70 hertz. So you will see a signal of gamma oscillations. So those gamma oscillations are also, the rhythm also generated by PV neurons. Those are excited by pyramidal neurons, and then they give out the rhythm by the network of those PV neurons. And if you keep filtering in a higher frequency, you see other power in the about 100 to 150 hertz. Those are ripple oscillations, are also generated by those PV neurons. And you can see that the amplitude become lower and lower when the frequency become higher. So one explanation is, so those network of PV neurons in the gamma oscillations, they are larger, a little bit larger. So the larger group will give a synchronized signal to the local field potential nearby. And in the ripple oscillation, those synchronized group are smaller, so that the power is less. And the outphased ripple oscillations are canceled each other, so that the overall amplitude are smaller in the ripple oscillation. So this is what in the ex vivo brain slides, what the sharp wave will look like. And usually we put an electrode in the striatum radiatum in the CA1 area, as showing in the left panel. So those are hippocampal slides. So you can see that there's a CA1 on the bottom is the dentate gyrus, and on the right is the CA3, and then there's a mousy fibers connect to CA1, and we put an electrode in the striatum radiatum where the mousy fibers and the distal dendrites are, so which gives a larger local field potential and also clear ripple oscillations. Actually, the ripple oscillations are larger if you put the electrode next to the soma in the striatum of pyramidal neurons, because those PV neurons are having those synapses surrounding the dendrites close to the soma. So the ripple oscillations will have a little bit larger amplitude there. Okay, So why this ex vivo shockwaves are important for brain degeneration research? So one is that we have studied degeneration in many systems. Like we see brain degeneration have a correlation with the behavior of the animal. We have lots of AD model animals with a particular gene or expression of proteins that will promote amyloid beta. And so that will make the behavior a little bit, or the memory of the a little bit worse. So, and also we tested we studied the degeneration in the cell cultures, right? The cell culture, we can put amyloid beta in the culture system and record their activity, record their effects. So on the sharp wave in brain slides, they're sort of in the middle. That's we call the population, where there's a circuits, local circuits, which is like a CA1 circuits, pyramidal and the PV neuron circuits, and the CA3 is highly recurrent excitatory connections that's generating the sharp wave. And then using the Schaefer collateral fibers to make the sharp wave express in the CA1 area. So we are in the brain slide, when we have a sharp wave, and those activities are at the population neuronal level, right? So it's like a system. And many drugs will affect more, so those population neuronal activity are more sensitive to the drug effect and maybe more sensitive to the degenerative effects, like loss of neurons or loss of dendrites, loss of synapse, modification of synapses, right? And the second reason I would say it's generating sharp wave need a perfect, very delicate excitation inhibition balance. We know that the brain have to have a very balanced excitation and inhibition, so the activity won't run away, and or if the inhibition is too strong, and then the input, or they don't have much activity, right? So like so the balance between excitation and inhibition are important. And the degeneration often affect the balance, and many compounds can help to restore these balances, are used for those brain degeneration effects. And the sharp wave itself is actually generated with a glutamate and the GABAergic neurons, and there are many receptors, and also neuromodulatory synapses or receptors like serotonin, like dopamine. Those are highly expressed in hippocampus, like acetylcholine, right? So if we have acetylcholine put in the hippocampus, the sharp wave will stop and then become theta oscillation. So those are like a in vitro ex vivo model of the two state, transition between the two state from awake state to sleep state, right? Sleep states are one with the sharp waves, and the awake states are the theta oscillations, and by the neuromodulator of acetylcholine. So giving those multiple mechanisms, so the ex vivo sharp wave ripples are sensitive to a large variety of neuroactive compounds. And those drugs, you know, we have those clinical drugs. Those are not purely affecting only one receptor or only one mechanism. Usually they have a multiple effects, some major effects, let's say for serotonin, and minor effects for dopamine, for epinephrine, right? So those drugs having those combined effects and will have a combined effect on system. And sharpwave, since it's a population neural activity in hippocampus and involve many mechanisms. So those compounds, the effects can be sensitively detected by ex vivo sharpwave ripples. So that we will say sometimes using the ex vivo slides can be more sensitive, the test can be more sensitive than behavioral cell, the test in cell cultures. Also, this sharpwave is spontaneously generated. It's generated by intrinsic mechanism, pretty much like a heartbeat. So the mechanisms affecting the frequency or the repeating rate are also important for the cognitive functions. So there are several papers about when the mouse is making a decision, and then those are you have a burst of a sharp waves ripples. So those, if you have those bursts and the rate for generating correct response or correct decision are higher, so that affecting the rate of generating sharp waves may make the animal smarter, right? Or make it less, make people less affected by brain degeneration. Okay, so the enhancing the sharp waves may improve hippocampal dependent memory, which is a major deficit of Alzheimer's diseases. All right, and those compounds affecting ex vivo sharp waves, I asked the ChatGPT that says estimated about 500 compounds. So I have a list. I don't have time. I don't want to read the whole thing, but those are compounds actually known to affect sharp waves. Okay. So now we will shift the gear a little bit and talk about how to get a good slice for to get a good sharp waves, the slides with a good sharp wave. So basically, you cannot see when you cut the slides with a vibratone or compressed tone, you cannot see this slice is good or bad. But if you put a local field potential electrode on, you can see that on the left top trace, the good slides have a higher amplitude. The sharp wave have higher amplitude and more frequent. And often you see a burst of several of them. And in the, we call the bad slides, they have a lower amplitude and fewer, less frequent sharp wave ripple events. So we sort of say good slides are with a higher amplitude sharp waves. So higher amplitude means a larger fraction of cells are following the sharp wave. And those cells are mainly pyramidal cells that generate large field potential signals. But of course, in the, if the larger fraction pyramidal cells are involved, the inhibitory neurons, also a larger fraction, have to be involved. And the rate is higher means the generating circuits in the CA3 is preserved. The lower rate means either you cut too larger fraction of Schaefer collateral fibers, or the circuits generating sharp waves are not well balanced. And also, good slides means no seizure-like burst. Later, I will show an example of seizure-like burst in the slides. So those inhibitory neurons in the hippocampus, about 15% or 20% are PV neurons. And those neurons are holding the pyramidal cells, not generating seizure-like burst, right? So those seizure-like bursts are, let's say in CA3, the pyramidal cells are receptorally connected. If you don't have enough inhibitory force, so those recurrent excitation will run away and become a seizure-like burst. But they are not like like epileptic activity, constantly have a episode of many spikes. They only have a one, but it's still a good indication that inhibitor neurons are not well preserved. Okay, so now how do we get a good slides from ex vivo brain slides? So and the Precisionary have a previous webinar by Dr. Jonathan Ting, which helped me a lot about those solutions and the steps for getting good slides. Mostly they will preserve the cells and circuits during the slicing and the recovery. So I won't repeat those particular procedures like particular solutions, EMDG, and antioxidant solutions for preserving it, and also the temperatures, several temperatures for slide recovery. So I will emphasize a little bit more mechanical on how to preserve Schaffer collateral connections. So Schaffer collateral connections are long axons on the cellular scale. So usually longer fibers are more difficult to preserve. And so this is one important thing that we use vibratome, use compressed tone, we can do better protection of Schaffer collateral connections. Another is maintain good variability to reduce calcium loading in the tissue and protect inhibitory neurons and eliminate spreading depressions. So spreading depression are also started with like excitatory neuron activity, and that they dump a lot of potassium into the extracellular space, and those potassium will keep more excitation and then become depression in the network. So with a balanced EI. All right, so let's do preserving Schaffer collateral fibers. So there was we call a magic angle. So this is like a 12.7 degree first, like proposed by a group of pioneers doing in vitro ex vivo sharp waves, particularly the paper by Mitsunuma 2014. So they explained this in more detail. Basically, you have a rostral high angle of 12.7 degrees. So that one will more parallel to the Schaffer collateral fibers in the hippocampal slides. Therefore, you can preserve more. And also, you want a relatively thicker brain slice. We usually do 480, 450, 480. So those, if you perfuse the slides two side, and those are deliver enough oxygen for sharp wave ripples. Okay, so this is a magic angle and a relatively thicker slides. And another one is a lateral sleeping damage. So if you use we use a storm light, certain frequency flashing light to observe the vibratome blade. So we find the brain in the middle, the brain slab moving laterally slip a lot, about a millimeter sometimes with the blade. So those are pretty bad for, they will pull or broken those long fibers. And the precisionary compressed tone is actually a genius invention. That's hold on the right, a hold give a compression, the piston give an upright compression, and those colors, maroon colors, give a circular compression so that the brain tissue no longer move with the blade. So we find, so with the compressed tone, the the quality of the slides are largely improved. And so those are with the brain tissue are holding. Here you see in the blue are low gelling point temperature agar. So that's actually hold the tissue not moving with the blade, with a compression force, circular compression force. So those are like a genius invention by Dr. Chu, I think some 20 years or 20, 30 years ago. All right. So now I'm talking about some small gadgets we made in our lab. So why we do that? Because I'm kind of training master students. So I train like 1 or 2 for recording sharp waves from ex vivo slices. I train 1 or 2 master students and sometimes 1 or 2 PhD students each semester. And those people, how can we make those new students be trained in a few weeks? Because they only have 1 semester and they want a project. They want to see, this compounds can affect sharp waves. How can we do that? So we make several gadgets in the lab, so make the slicing easier using compressed tone. And before that, people, most of the students would prefer ordinary vibratone because the vibratone is easy, right? So you put a brain slab with a glue on the stage and then you cut. It's easier. But for compressed tone, you have to glue the brain tissue on the piston, and then you have to do the low temperature agar gelling, and there are several more steps, and those will make students feel more complicated, and then they would not prefer using it because it take a longer time to learn. So for this purpose, we sort of make one in the middle. It's a holder for the piston, and we cut the piston with the magic angle 12.7, and then the holder will adjust the angle so that the surface, the cutting surface of the piston is parallel to the floor. And then we put a brain onto a petri dish, just put them on, and so that on the petri dish, you can rotate and move the location of the tissue and also the angle, the rostral caudal angle of the tissue to orientate, fit correctly with the oblique cutting angle of 12.7 degrees, the rostral high angle. Then, so this one actually break up those steps into several simple steps. And so actually, the student will like to use it because each step is relatively easy. And on the right top, so this device mostly give you a correct oblique angle of the cutting, and also we have a micro drive on the top. It provide a gentle touch when you glue. So the glue is applied to the surface, and then you turn the holder down, so the glue will be there, and then you use a micro drive to gently touch the piston to the tissue, and with a little pressure, so that it will glue well. And then we found the glue failure is the most majority factor of fail to get a slides, good slides with good sharp waves. Mostly when the tissue is in the wrong angle, and then the sharp waves are all ruined afterwards. So the second thing we had is a worm block. It's a piece of metal, and we have a little heater, a temperature controlled heater, to make it a 36 degree. And we can use anything 36 degree, like a water bath, for this purpose. So because the agar, low gelling point agar, you have to melt it with a high temperature. And then after melting the agar, if you quickly pour it into the color and with the tissue will be cooked by this high temperature. So it's important to cool those agar, liquid form of agar to 36 degrees, which they are still stay as a liquid, and then but not damage the tissue. So those heating blocks are used for this purpose. And you put a high temperature agar, 90-degree melted agar into this block and keep it for 10 minutes. And then those agars will cool down to 36 degrees and then will not damage the tissue. And also you can keep the agar liquid in the liquid form for a long time. So for students who are not familiar with the procedure, if you don't have this and people are rushing to pour, and then sometimes the gelling will be premature. They were already gelled before you pour into it, or you rushed for other procedures with those psychological pressure and cause some more errors. And those warm block will reduce all these factors for failing to cut good slides. And third thing, which we found that's pretty useful, it's a small telescope. Those are telescopes where we just use a projecting lens from old movie projector, but any small telescope with a 2-piece of lens should work. So this one will have a focus point. We'll see the edge, the edge between the blade and the color, and to adjust the edge lower than 1 millimeter. So we are using the old version of compressed tone, but I heard in the new version, the distance automatically adjusted. So we don't really need this telescope. But we still use that to see if there's a uneven push, which means so those agars are prematurely gelled in the piston, so that when you push them, and the angle of the tissue would change a little, and with those telescopes you can clearly see those uneven push. Also, sometimes the cell tissue are damaged before you glue it, and then when they cut, you see a cloud of cells coming out from the tissue, and that will estimate the damage when you dissect the brain, take them out from the scalp, or other problems in the solution failure. And for those cutting, we all use standard high sucrose cutting solutions, and to simplify the procedure. And after cutting, we're moving this in one step into 32-degree normal incubation solution, just to simplify the procedure. And we found those are simplified steps are good enough to get a good sharp waves from the ex vivo hippocampal tissue. Okay, so now I'm showing a slides from GCAM6 labeled, transgenetically labeled animals. So you can see on the left, there's a hippocampus section, and with several areas with a higher fluorescent intensity. So those mousy fibers are somehow very bright, and mousy fibers actually label the area of CA3. And also on the middle and the right, we see a band of high fluorescent area. So those are not quite following the anatomical structures, but those high calcium loading are probably most likely related to the cutting. So you see those cutting this slide was cut by a vibraton, Leica vibraton. So those cutting areas are, for no reason, we don't know quite know what's the reason, but you see a band of high calcium loading in the cells, from DG all the way go up to the CA3 area. And the calcium loading usually don't get a very good sharp wave signals. And third high calcium load area is surrounding the electrode. So if you advance the electrode not too carefully, you create a little micro damage to the tissue. And all you care with high amplitude sharp waves are from the tissue surrounding the electrode. So when you put the electrode in, you create a little damage, and then the signal, the amplitude will be small. And on the right, those dots are the amplitude of sharp waves, and the x-axis are time, seconds. So when we first put the electrode, often we see the amplitude was reduced, and it takes about 20 minutes to recover to 90% of the normal amplitude. So those are indication of damage just surrounding the recording local field potential electrode. And those are actually affecting our research a lot, because you need a long period of recovering time. So on the left image, you can see those electrodes create an area of higher calcium loading. So those are also another indication of damaged tissue just by the electrode, micro damage by the electrode. Okay, so I don't have much time. Whoops, I just gave you a few research examples about calcium imaging of sharp waves. So, but for the calcium imaging, it's like two kinds of calcium GCAM signals. One of them is the cellular signals. You see blinking cells. You can put a slide under a 2-photon microscope. You see those cells are blinking. However, when those slides having sharp waves, the blinking doesn't have a good correlation with the hippocampal sharp waves, with the field potential sharp waves. The reason is on each sharp wave, they have only 5% pyramidal cells synchronized to the field potential. So that when you see many cells, and those membership of those 5% are changing from sharp wave to event, and then you don't get a very good correlation. There's another population calcium signals, which are have a one-to-one correlation with the field potential. Those are population calcium signals on the right. So you can see those blue trace are blue, and the red trace are calcium signals from two spots. And they are correlated pretty well with the field potentials. And you look at a scale bar, so those are pretty small signals. The delta F over F are only 0.5% of the resting calcium signal. It's about resting fluorescent intensity, only 0.5%. And the cellular signals, usually 20% or even higher, or sometimes 200% change in the resting light. So the population calcium signals, we cannot use 2-photon to see them, because 2 photons are too noisy in the low fractional change rate. Use ordinary camera and ordinary fluorescent microscope, we can pretty clearly see those calcium signals during the sharp wave. And the advantage being, when you have a optically recording sharp waves, we don't have the period of recovery period of electrode. So another example I will show is optically recorded sharp wave epileptic events. So during this recording, there's a spontaneous, like a single event of an epileptic runoff event. So those are didn't generate large signal in the field potential. They generate huge signal in the GCamp sharp wave signals, which is about sometimes are like a 10 times larger. This is because the sharp waves have only 5% parameters are in sync with the field potential, and interequal spikes are much higher. Sometimes 40–50% of cells will fire in a short period and generate huge calcium signal. So the third example will be attenuation of ETTAs. ETTAs are excitatory like a transporter, calcium-dependent glutamate or transporter, amino acid transporters in the glial and in the neurons. So those are important to keep the ambient extracellular glutamate low. And we know that amyloid beta actually disrupt those ETTAs. And so we put a small molecule drug, TBOA, which actually block the ETTAs, those transporters. And we found it has a huge effect on the sharp wave ripples. So they basically disrupt the CA1, reduce a lot the CA1 sharp wave amplitude. And this effect is reversible, so you can washed out. So this become a artistic system to study how can we mitigate this ETTA attenuations. All right. So the third example will be try to manipulate the sharp wave occurrence. So we use a tiny electrical field stimulation to pace the sharp wave. And the pace the sharp wave, you see the trace on the right, pretty similar to the spontaneous sharp waves. They have a variety of amplitude, not dependent to the stimulus intensity. And on the bottom chart, the distribution chart, you can see the pacing rate generate a peak in the sharp wave generation. So that way, you can effectively increase the rate of sharp waves. We know that in vivo, if the sharp waves can be generated, more sharp wave or make it a prolong the burst of sharp wave, that will make the animal making better, smarter decisions. However, those are if you use the electric field to try to promote the sharp wave, in slide it works, but in vivo it didn't work. So this is still an open question. How to pacing the sharp waves in the hippocampal slides. I'm sorry for being too long, and so this is my conclusion and about my talk, and I will recommend compressed tone if you want to do study sharp waves in ex vivo slides. All right, thank you very much. Those are the people who did the work I mentioned in the talk.
Professor Wu, thank you so much for your talk today. On behalf of Precisionary, one of the most exciting things we have learned from you is the ability to embed your tissue with ease using this apparatus that you have developed, which accounts for the embedding angle and also having two hands free. And we're so happy to see that your students learned it so quickly, and we hope to adapt that so that it's available for everyone. And we hope to involve you in that process as well.
Thank you.
In the Q&A, I don't have any questions, but I'm going to go ahead and open up the floor, and I'll be able to hear any questions for speakers or attendees who want to speak. And if not, as usual, we have a lot of shy attendees. They can always reach out later. And so with that, Dr. Wu, I want to thank you again. And I hope everybody here has a great week. And thanks to all of our attendees as well.
Great. Thank you very much for the opportunity.